Welcome to our presentation on the cell cycle. A couple of key concepts here. Cells have distinct phases of growth, reproduction, and normal functions. Here's a picture of a cell just about to finish dividing. The late stages of cytokinesis. The cell has four main stages. The cell cycle is a regular pattern of growth, DNA replication, and cell division. Usually it's shown as a circle because it's a never-ending thing. It cycles and cycles. The interphase is the parts gap 1 or G1, S and gap 2, G2. It's called interphase. And then the mitosis and cytokinesis phases are the areas where the cell is actively dividing. Main stages are, as we said, G1 or gap 1, synthesis or S, gap 2, G2, and mitosis. Gap 1, G1 is normal cell growth and function. Just the cell acts the way it's supposed to. It's a hair cell, it's a hair cell. Skin cell, it acts like a skin cell. In S, or synthesis, is the DNA replication phase. It's when the DNA is copied. It's starting to, something has kicked the cell into a cell division mode. Gap 2 is additional growth. We need to get more organelles. We need to prepare the cytoplasm. S is preparing the nucleus for cell division. Gap 2 is preparing the cytoplasm for cell division. And then the M phase, with prophase, metaphase, anaphase, telophase, are the four steps in nuclear division. The M phase is when the nucleus divides, and then finally cytokinesis is when the cytoplasm divides. Mitosis only occurs if the cell is large enough and the DNA is undamaged. Cell division happens at different rates. The rate of cell division varies for the types of cells. Here's just a chart. Skin cells reproduce pretty rapidly. You pretty much every 2 weeks or so end up with new sloughing off old dead cells and new ones. Red blood cells are 4 months, and so you can read the chart. Different things can be longer, and I'm not listed on here. In nerve cells, in some cases, is believed that they never divide once you've become fully grown. So they would not go through the full cell cycle. They'll be stuck in G1 forever. The cell size is limited. It can only get so large. If it starts to exceed that, it has trouble servicing itself from the outside. The volume increases faster than the surface area. The internal area is the volume. It increases faster than the surface area, and that's the area where things get moved out of the cell or moved into the cell, the surface area. So if the surface area relative to the volume is decreasing, you're not able to bring good things in and bad things out at a fast enough rate as the volume increases. And so there's a practical size limit to cells. So you see here we have just a little chart kind of showing that. 1, the surface area is 6, the volume is 1, so the ratio is 6 to 1. That's a pretty good surface area to volume. And double it in size, then it's 24 to 8. That's a 3 to 1. We've gone half. And if we go 3 times bigger, then it's the surface area is 54, the volume is now 27, and it's 2 to 1. So now we're really getting down there in the amount of surface area there is to service the volume. So then the only thing you can do is to divide and get back down to a smaller size. Surface area must be adequate for the exchange of materials. I was just saying, good things in, bad things out, and that's coordinated by division. If a cell gets too big, it kicks into a mode of cell divisions. This is just a couple of pictures of cells here. Cells that must be large have unique shapes. There's a blood cell in the lower left, nerve cell, and I believe some skin cells there. So